To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.
Hello my dear students, welcome to today's lesson. So I am going to continue the lesson inheritance. In the previous chapter, we looked at what inheritance is. All living organisms, plants, animals, us, we inherit characteristics from our previous generation. And under that, you all were able to understand that some characters go from one generation to the other, whereas some characteristics can skip a generation. So, character that is seen in your grandparents will not be present in your parental generation, but it can be seen in your generation. So, like that also characters can be inherited. And thereafter, we looked at different types of characters. Common inherited characters, which are very common among the human population. You all can remember we looked at different characteristics. The complexion, color of skin. Then we looked at the type of hair, straight and curly hair. Thereafter, we saw the nature of the earlobes, whether they are fused or free earlobes. And then the ability to fold the tongue. Dimples on your cheeks. Thereafter, the widow's peak and also how the thumb will be positioned when you cross your fingers, as well as straight and curved thumb. So, those are some common inherited characters. Thereafter, we discuss some of the rare inherited characters. So, you all can remember rarely inherited characters. What are they? They are not very common among people, but they are there and they are inherited characteristics. So, as examples, we looked at syndactylity, where the fingers or the toes are fused together. Then polydactyl, where additional fingers or toes are present. Then albinism, no pigmentation, hair, the skin, everything is white in color. And also color of the iris, color of the eye, brown or blue eyes. So those are rare inherited characters. You would have seen people with these characteristics, but very rarely. Then we discuss the characteristics that are not transmitted from one generation to the other. Non-inherited characteristics. Due to environmental changes, lifestyle, activities, exercise, if you can build up your muscles, if the muscle grows due to these factors, that is not inherited by the next generation. So now you all have a very good understanding about all these characteristics. And also I told you all, Gregor Mendel, he is the father of genetics. Why? Because he was the one who did a lot of experiments related to inheritance. So that is what we are going to discuss now. So you can see here Gregor Mendel, father of genetics. The first person to study about the transmission of inherited characteristics. So here transmission of inherited characteristics. How these characteristics go from one generation to the other, or whether they skip the generation, all those are studied under transmission of inherited characteristics. And he is an Austrian priest and a science graduate, Gregor Mendel. So you have to remember his name, Gregor Mendel. And he is honored as the father of genetics. So, very important because he is the person who carried out a lot of experiments related to inheritance and he explained what inheritance is. Although he did not have the knowledge of DNA, genetics, those knowledge were not there because some of the technology was not developed, but still he was able to explain inheritance. So, that is why we call him or we honor him as father of genetics. Gregor Mendel. Now we will see how he did the experiment and what type of plants he used for his experiments and the reason for selecting that plant. He actually selected the plant Pisum sativum. Pisum sativum is the garden pea plant. So it has so many desired characteristics that made him select the garden pea plant as the plant for his experiment. So with that, we will move on to the next part of the lesson. Mendel's experiments about inheritance. So he used the garden pea plant. Like I told you all, the garden pea plant, it's very important. Now here I am underlining these students to highlight what is needed for you all. But you can see Pisum sativum, this is the scientific name. You all know the rules that we need to follow when we need to write the scientific name. What do? It has to be written in Latin or the English name, generic epithet, specific epithet. Generic is Pisum, specific is sativum. And when it is printed, it will be italicized. So that is done here. Only when it is handwritten, we have to underline them separately. The two epithets have to be underlined separately. So here what I have underlined is to show its importance, not part of the scientific nomenclature. You all have to remember that. And also always we need to have a space between the two epithets. Those are the rules that you need to follow or conventions that you need to follow when writing scientific name. And here you have to remember the scientific name Pisum sativum, that is the garden pea plant. So here you can see the garden pea plant. You can see the pods. We discussed pods if you remember in the previous chapter also. This is a pod. Inside the pod you get the seeds. So these are green color pods. You can see the leaves and you can see a white color flower there, a small white color flower there. So this is the white color flower, then the pod. So these are all external features that are used to identify the garden pea plant. And we said the external features are also inherited from one generation to the other. So that's a garden pea plant. So he used, that is Mendel used the garden pea plant, that's Pisum sativum plant, for his experiments in 1865. He has used that particular plant for his experiment because of its special features. So I told you all there are many special features. Now if you look at this, you can see the pod, green color pod. Then you can see a white color flower. Another type of Pisum sativum, it has a purple color flower. So between purple color flower and white color flowers, the difference is very distinct. You can easily identify the difference. If it was white, off-white, then kind of a light yellow, then it's difficult to differentiate the colors of flowers. But here, white and purple, the colors are very distinct. The same way, if you look at the size of the plant, there are short plants, tall plants. There are no medium heights. In between heights are not there. Short, tall. So very distinct features. Then there are round seeds, wrinkled seeds. So like that, this particular plant has very distinct characteristics. So not only that, students, there are so many other features that made Mendel to select the garden pea plant for his experiment. So in the next slide, I will discuss those characteristics. The reasons to select the garden pea plant for his experiment are as follows. The first one, it can be grown very easily. So when we say it can be grown very easily, you don't have to give any special conditions. In the normal fertile soil in a garden space with the normal requirements for sunlight, air, water, nutrients, you can easily grow the garden pea plant. So that is an advantage. You don't have to have a special technique or you don't have to provide special conditions. So that is the first reason. Then the second one, can obtain the yield within a short period of time. Now you all have heard about annual plants. Plants that have a life cycle of 1 year. Annual means year. So 1 year. The life cycle of garden pea plants is 1 year. So within 1 year, you can grow the one plant generation. It will reproduce, it will produce the fruits, and then the next generation will continue. So within a short period of time, you can get the yield. So you can see what is being produced. Within a year, you can see the color of flowers, you can see the nature of fruits, all that can be seen. So that is also very important. Now if it was not an annual plant, if the life cycle takes about 10 years, so to get the observation, you have to wait for 10 years. If you want to observe the second generation, you have to wait for 20 years. So that's a very long period of time. So those plants are not very suitable for these types of experiments. So here the garden pea plant, that is Pisum sativum, is an annual plant. Within 1 year, the life cycle is completed. So because of that, the yield can be obtained within a short period of time. The whole life cycle is 1 year. So before that, you will get the yield. That is the second reason. So first one, can be grown easily. Second one, can obtain the yield within a short period of time. Then the third one, can obtain pure breeding plants. Now when we say pure breeding plants, this is what it means. Now you have tall plants, you have short plants. Now this tall character is only tall character. If it is short character, it is only short character. There is no mixing of characteristics. So that is known as a pure breed. Now in dogs, cats, you would have heard about this term. Pure breed. If it is a German Shepherd, it is a pure breed dog. So here the dog's parents, the grandparents, all are German Shepherds. No mixing of characteristics. If you cross a German Shepherd with another type of dog, then it is a crossbred one, not a purebred one. So pure breed means it only has a certain characteristic, pure characteristics. So the Pisum sativum has only the characteristics of Pisum sativum. It is not crossed with any other plants. The characteristics are not mixed with any other plants. That is pure breeding. So here you can see, can obtain pure breeding plants. That is, the selected characteristics are not changed for many generations. So the tall plant goes on as tall plants for many generations. Short plants become short plants in the next generation and the generations after as well. But if it is a mixed character, then what can happen is the tall plant can produce short plants also. Then it is not a pure breed. So that is how we identify. If it is only the same characteristics that are carried out for generations, it is pure breed plants. If the characteristic changes, then it will not be a pure breed plant. But Pisum sativum, there are pure breeding plants. Then the next characteristic, presence of easily observable contrasting characters. So that is also important. Easily observable contrasting characters. So like I told you all, tall and short plants, you can differentiate easily because there are no intermediate heights, either short or very tall. Then yellow and green seeds. So again, two different colors. Yellow, green, obviously you can differentiate the colors. If it was like I told you, light yellow, dark yellow, medium yellow, it's difficult to differentiate. But here, yellow and green, totally different colors, the seeds. Then tall and short plants, like I told you all. Similar to that, I told you all, purple and white flowers, two different colors. Then round seed, wrinkle seed. Round is a completely round seed. Wrinkle, the shape will be different. So like that, there are very contrasting observable characteristics. Tall and short are contrasting. Yellow and green are contrasting. Round and wrinkled are contrasting. Then white and purple flowers, that is also contrasting characteristics. So that is again another reason why he chose garden pea plants. Then the next one. Naturally self-pollinating. So normally, you know what pollination is. What is pollination? The depositing of mature pollens onto the stigma of the flower. So that can happen in two ways, self-pollination, cross-pollination. If the pollens and the stigma are from the same flower, there is only one flower, then we call it self-pollination. And in self-pollination, there is no mixing of characteristics. So now can you all relate the other features? There is only one flower, self-pollination occurs. So let's say a tall plant. Flower of the tall plant, self-pollination occurs. So there is no mixing of characteristics, no variation, no new plants are produced. Therefore, you will get the pure breed tall plants in the next generation. So now you can relate the features. So naturally they are self-pollinating. Same goes for short plants. You have the short Pisum sativum plants. They undergo self-pollination. So no variations, no new characteristics. All the generations will have short plants only. So you will get pure breeding plants. But there is the second type of pollination. What is that? Cross-pollination. Cross-pollination involves two flowers. Mature pollens from one flower is deposited on the stigma of another flower belonging to the same plant or of the same species, another plant of the same species. So either way, there are two flowers involved. Then of course, there is a chance of mixing of characteristics, variations, so new characteristics can be formed. So when these characteristics mix and new characteristics are formed, you can get pure breed plants. In addition to that, you will get cross-breeded plants, mixing of characteristics. So that is also possible with this Pisum sativa. So here, naturally self-pollinating, but when necessary, cross-pollination is also possible. So normally it carries out self-pollination. So no variations, pure breed plants. But if you want, you can make cross-pollination to occur. So you are familiar with that also. Pollination techniques. Normally there are agents of pollinations. You all can remember that. The insects, then wind, water, all these are agents of pollination. But we can carry out artificial pollination. What do we do there? You can use your fingers to get the pollens and deposit them on the stigma, or we can use a paintbrush, or you can use forceps. So there you take the pollens from one flower and deposit it on the stigma of another flower. So cross-pollination is possible whenever necessary, and you all are familiar with the techniques also. Then the last reason, ability to obtain offsprings by breeding and can continue the generations. Now this is something you have to understand. Ability to obtain offsprings by breeding and can continue the generations. Now what is the meaning of breeding? When we say breeding, we are trying to modify characteristics so that we get characteristics that are more useful to us. So there basically it is the cross-breeding of plants. You can mix characteristics and you can try to get the desired characteristics using genetics. So that is the concept of breeding, getting characteristics that are useful for man. So there, those breeded plants, crossbreeding can happen. So those breeded plants also can continue the generation. So new characteristics can be continued from generation to generation. Can you all understand all these? Now you have the garden pea plants. They have distinct characteristics. Their life cycle is short, so you can look at the yield. They are pure breed plants because they normally do self-pollination. But if necessary, we can carry out cross-pollination, or we make them to undergo cross-pollination, and also breeding can be carried out. And the breeded characters, the new characteristics also can continue from generation to generation. So this is an ideal plant. You can observe all types of inheritance. So here Mendel was actually able to observe many things related to inheritance because of all these features or the reasons. So you have to remember them. The reasons to select the garden pea plant for his experiment are as follows: can be easily grown, can obtain the yield within a short period of time, can obtain pure breeding plants, that is, the selected characteristics are not changed for many generations, presence of easily observable contrasting characteristics like a yellow-green seeds, tall short plants, then naturally self-pollinating. When necessary, cross-pollination is also possible, and also ability to obtain offsprings by breeding and can continue the generations. And I am sure you all are familiar with the term offspring. Now, if you have the parents, the children are offspring. So, likewise, you have the parent plants. The new plants that are produced from reproduction are the offspring. So, offspring is a term that is used to refer to the new generation that is formed from the parental generation. So, it can be plants, animals, or even people. That is the meaning of offspring. So, these are the reasons for selecting the garden pea plant. You all have to remember these reasons, students. Pisum sativum was selected by Gregor Mendel for his experiments, and these are the reasons for selecting that plant. I'm sure you all can understand that clearly. So with that, we'll move on to the next slide. So he observed 7 contrasting characters. So 7 contrasting characters. Now you all know the meaning of the contrasting characters. Tall, short, yellow, green, white, purple, round, wrinkled. All these are contrasting characters. Very distinctly different characters. So he observed 7 contrasting characters. That's important. You have to remember these 7 contrasting characters and tested 1 character at a time. So that's also important. He tested only 1 character at a time. Although he observed 7 characteristics, he didn't look at all 7 characteristics together. He did the experiment separately. So, if he observed the tall and short characteristic of a plant, he did not observe the yellow and green seeds. Or when he was observing the color of the seed, he didn't consider the tall, short character of the plant or the color of the flower. So, although he used 7 characteristics, he took the characteristics separately. So, 1 characteristic at a time. So, because of that, this is given the name monohybrid cross. We will be discussing that again also. Mono means 1. So, 1 character at a time, and it is a hybrid, mixing of characteristics. So, crossing the plants, crossbreeding the plants. So, for example, if we took the tall and short characteristics, that is only 1 character, height of the plant. Although they are distinctly different, tall and short are different, but both tall and short refer to the height of the plant. So, it is only 1 character. There, he did the crossing of plants and he observed the generation. So, we will be referring to this as monohybrid cross. Again, we will come across that same term in the one of the next slides. So what he did was, the procedure he followed for the tall and short character was as follows. So this is what he did when he considered the tall and short character. Before reading this, I will explain the procedure and then we will go through the explanation. So what he did was, he had parent plants, tall plants and short plants, and then he crossbreeded them. Between the tall and short plants, he carried out crossbreeding. Crossbreeding. Because normally it is self-pollination. But here this was crossbreeding. So cross-pollination. What he did was, pollens from the tall plants were deposited on the stigma of the short plants, whereas pollens from the short plants were deposited on the stigma of the tall plants. So complete crossbreeding by carrying out cross-pollination artificially. Pollens from tall plants were deposited on the stigma of the short plants. Pollens from the short plants were deposited on the stigma of the tall plants. So complete crossbreeding. He selected the parents, then he made them to undergo crossbreeding. From that, he got the first generation. First generation, that is denoted as F1 generation. When we say F1 generation, it is called first filial generation. First filial generation. So when he did this crossbreeding, he saw in the first generation, there were only tall plants. Only tall plants. That is also important. Only tall plants. Now here students, I am giving you all the explanation. That is why in between I am writing all this. But after you understand the procedure, there is the correct way of showing how the generations are formed and characteristics are inherited. So here I am writing additional information as well for you all to understand the procedure. So first generation was all tall plants. Then what he did was, he took the tall plants as parents. So these tall plants were considered as parents. And then what he did was, he allowed them to undergo self-pollination. Here there was self-pollination. So what can you see there? Tall and short plants were the parents. They underwent crossbreeding. All the plants that were formed were tall plants. No short characteristic. So one characteristic was missing. Then what he did was, he took them as parents and led them to undergo self-pollination. So thereafter, he observed the second generation. Then he observed the F2 generation. In F2 generation, again there were tall and short plants. There were tall and short plants. That is, after the first generation underwent self-pollination, there was tall and short plants. So what can you see there? Earlier tall and short parents were there, pure breeds. But the first generation was only tall plants. And when they underwent reproduction by self-pollination, there were tall and short plants again seen in the second generation. Now you all can understand the concepts we discussed before. Now the short character was there in the parent generation, but it was not seen in the first generation. It was again observed in the second generation. So the short character did not go from one generation to the other. It went from the parent generation to the F2 generation. And there is a reason for that. That is what Mendel explained under inheritance. This was the observation. And there's one more thing. Now we will discuss how that ratio is obtained. Always, when he carried out this experiment, between the characteristics, there was a ratio. In the second generation, the ratio was 3:1. So let's say if he had 4 plants, 3 plants will be tall and 1 plant will be short. If he had 400 plants, almost 300 plants will be tall and almost 100 plants will be short. But it is not the exact value. It can be 298:102. But when you finally get the ratio, he saw that the ratio was 3:1. 3:1. Tall is to short. Every time he carried out the experiment between tall and short plants, the second generation, when he followed the same procedure, the second generation had this ratio. Tall and short characteristic. Those were the observation. Now, not only for this character, when he used the other characteristics, let's say round and wrinkled seeds, they are also same thing. He took the round and wrinkled seed plants, pure breed plants, led them to undergo cross-pollination, crossbreeding, and he got all round seeded plants, and then he led them to undergo self-pollination, and thereafter in the second generation, again he got round and wrinkled seeds, and the ratio between the round and wrinkled seeded plants was 3:1. So like this, he observed for all 7 characteristics, all 7 contrasting characteristics. Again, I will discuss the observations he got for all his experiments in one of the slides after this. So now you all have to understand the procedure clearly. We'll go back and so he observed 7 contrasting characters and tested 1 character at a time. The procedure he followed for the tall and short character was as follows. So what I explained. Cultivation of pure breeding tall and short plants. So he had pure breeding tall and short plants. These parental generation was known as P. The parental generation was known as P. I didn't use that letter there, parental generation P. Then pollens of tall plants were deposited on stigma of short plants and vice versa. So pollens of short plants were deposited on the stigma of the tall plants. So that was the cross-pollination what I told you. Obtain new seeds by cross-pollination and plant them to obtain the next generation. So from the parent generation, cross-pollination was carried out. After reproduction, the seeds were collected and planted to get the next generation. That is the first generation. All plants were tall. All plants were tall and they were referred to as F1 generation. What I told you. First, filial generation. F1 generation. Then, allow self-pollination to take place within F1 generation. The resultant seeds were planted to obtain F2 generation. And the tall to short plant ratio was 3:1. So this is the ratio, very important. Tall to short plant ratio was 3:1. Is that clear now? So this is what happened there. So now parents, it was referred to as P. Then tall and short plants, crossbreeding, the seeds were taken, they were planted, he got the first generation. All were tall plants. Then after that, he allowed them for self-pollination and got the seeds, planted them, got the F2 generation and got the ratio as 3:1. Similarly, he carried out the experiment for all 7 contrasting characteristics. That was Mendel's experiment. I'm sure you all can understand that procedure clearly now. So now we will move on to the next slide. It was a question to Mendel that what has happened to short character in F1 generation. So that was a question. What has happened to short character in F1 generation? Because all the plants were tall plants. So like I told you all, the short character was seen in the parental generation, pure breed short plants. That was not seen in the first generation, but it again reappeared in the second generation. According to Mendel's opinion, the tall character was dominant and short was recessive. That's again an important thing. Tall character was dominant and short was recessive. Now you had the tall plants, short plants. This tall character, because it was dominant, it dominated the short character. It did not allow the short character to be expressed in the first generation. So that is why all the plants were tall plants. It was possible because there was cross-pollination. Initially tall pure breed plants, short pure breed plants. But when they were cross-pollinated, the tall character that was dominant suppressed or did not allow the short character to be expressed. So that means the short character is known as the recessive character. Recessive character. So tall, the character that is expressed in the first generation, is the dominant character. The character that was not expressed in the first generation is known as the recessive character. So we are observing the height of plant, 2 distinct characteristics, tall and short. In the first generation, only tall plants are obtained. That means the tall character is a dominant character. Short character is a recessive character. And always, in Mendel's experiment, the dominant character was expressed, or all the plants showed the dominant character in the first generation. The recessive character was not observed. That is how Mendel explained his observation, and that is what happens in inheritance also. Then, the feature which was recessive in F1 generation reappear in F2 generation. That is also something we saw. The feature that was recessive, it reappears in the F2 generation. It was an important observation, as Mendel used a single pair of contrasting characters at a time. It was known as monohybrid cross. I mentioned this term to you all. Monohybrid cross. Mono means one, hybrid is mixing of characters, and crossing also cross-pollination of plants, crossing the plants. So monohybrid cross. Since he only considered one character at a time. Is that clear to you all? One pair of contrasting characters. A single pair of contrasting characters. So we call it monohybrid cross. What is the meaning of single pair of contrasting characters? Tall and short is a pair of characters. They are contrasting but they both refer to the height of the plant. So, single pair of contrasting characters. Tall and short are single pair of contrasting characters. That is another term. Single pair of contrasting characters. So, because of that, we call it monohybrid cross. So, I am writing all these terms. They are all given in the explanation, but these are new to you all. You have to remember these terms. So, when you answer questions also, you have to use the exact terms. Dominant character, recessive character, parent generation, F1 generation, F2 generation, then single pair of contrasting characters. Because of that, we call it a monohybrid cross. I am sure up to now you all can understand this procedure. So with that, I will move on to the next slide. So there is an assignment that you all have to consider. Mendel has used large number of samples and repeatedly done the experiments to confirm his conclusions. Analyze how Mendel has carried out his experiment according to scientific method. So, you all are familiar with scientific method, students. What is the scientific method? You have a hypothesis. Before that, you observe something. So, actually, now Gregor Mendel was a priest and he was a scientist. So, he carried out experiments. He wanted to do certain things where he wanted to actually cross two colored flowers and get a new color. So, he carried out the experiment, but he got a different observation. So then what he did was, because he had this observation, he came to a hypothesis. He proposed a hypothesis. And based on the hypothesis, again he designed his experiment. So in that design, he decided to use pure breed plants, what we discussed before, like tall and short, or round and wrinkle seeds, white and purple flower. Like that, he used different distinct characteristics. 7 pairs of distinct or contrasting characteristics. So, from that, he carried out monohybrid crosses. But for one characteristic itself, for tall and short plants, he repeated the procedure several times. He did not just stick to one set of observation. And in each activity, each experiment, he used a large number of plants. Say 1,000, 2,000 plants, like that he used a large number. Because you all know in scientific method, when you use a large number of observations, you will get a more definite conclusion than just doing 1 or 2 activities. If you have many samples from all the observations, the final conclusion that you can obtain will be more specific. That is what happens in statistics also. Let's say when you have 2 colored marbles in a box, when you are drawing out marbles, the probability of getting one color, say red and black marbles, probability of getting red color. To find that, if you just draw out 2 marbles, that's not enough. Instead, you can draw out 20 marbles, 20 observations. It will give you a more better observation. If you repeat it 200 times, it's even more better. 2,000 times, it will be even more better. Even a coin. You all know when you toss a coin, the probability of getting head and tail is 1:1. 1/2 into 1/2, because the observations, there are 2 observations, head and tail, equal probability, equal chances of getting a tail and a head. But if you only do it twice, if you toss the coin only twice, sometimes you will get the same thing. You might only get the head. So you cannot come to a conclusion. But if you repeat it 20 times, 200 times, 2000 times, even 10,000 times, you will get the correct probability. So the more number of times you repeat, better observation you will get. So that is why he used large number of samples and he repeatedly did the experiment. So what they want you to do is, they want you to look at the procedure he used for his experiments and how he repeated it so many times so that he can come to the conclusion. And remember students, when Mendel did the activity, when he did the experiment, when he explained the inheritance process, nobody knew about genetics, genes, DNA molecule, nothing was known to them. All those were developed lately. After Mendel explained inheritance, long time after that only, they were able to observe the DNA molecules. So he did not have any knowledge about genetics, but from his experiment, because he carried out the experiment according to the correct scientific method, he was able to observe them properly, he was able to record the observations, and he was able to give a very good explanation. That is why we still call him the father of genetics. So up to now, we have discussed who Gregor Mendel is and that he carried out experiments and he explained the inheritance process. For that, he chose the garden pea plant, that is Pisum sativum plant, and he observed 7 pairs of contrasting characteristics, but he carried out the experiment considering one characteristic at a time. So that is why we call it as monohybrid cross. And during the monohybrid cross, he used pure breed parental plants. Then he allowed them to undergo cross-pollination or made them to undergo cross-pollination, obtained the seeds, planted them, got the first generation F1, first filial generation. Then he observed that. After that, he made the F1 generation to undergo self-pollination and then obtained the seeds, planted them, and he observed the second generation. From that, he explained inheritance. But after using a large number of samples and repeating the experiment many times. So that is how he became the father of genetics. So with that students, I am going to end this chapter, and in the next chapter, we will see and we will discuss how Mendel got the observations, or actually the observations of Mendel's experiments, and from there we will try to explain how inheritance takes place.
To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.